the last video, we had discussed about cell signaling, that's the communication of cells. Cells don't use the language that we speak, of course, but cells use a language involving signals. Yeah, in this video, we shall discuss the various signals in cell communication. So signals are basically molecules, atoms, ions, but signals can also be physical signals like light, temperature, voltage, pressure, etc. This reminds me of the senses that we have: vision, hearing, touch, taste, and smell. All these are also signals. These were traditionally called senses, but these are signals for the central nervous system. Now there can also be mechanical signals, forces exerted on or by the cells. Cells in a population can sense the density of the population and other parameters. Now let's move on to chemical signals. These are molecules or atoms that act as signals. They are also sometimes called ligands. It can be a peptide or amino acids or steroids, hormones, terpenoids, lipids or phospholipids, proteins or glycoproteins, etc. In fact, any molecule or atom can act as a chemical signal and trigger a response. Okay, so if you want to know what are terpenoids are responsible for the yellow color in sunflower, red color in tomatoes, and they are responsible for the aroma of eucalyptus oil, flavor of cinnamon or cloves or ginger. And terpenoids are also responsible for the production of steroids and sterols in animals. Now coming back to the topic, any molecule can act as a signal for the cell, whether it's a food material or pathogen associated material, oxygen level or NO level, ion, pharmaceutical drug or otherwise. Now let's classify these chemical molecules or ligands. They can also be called as ligands into two types. Molecules can be hydrophobic ligands or hydrophilic ligands. Now what is hydrophobic and hydrophilic? If a molecule is soluble in water, that it likes water, it is called hydrophilic here. Or if a molecule hates water, it doesn't like water, then it is called hydrophobic. It has a phobia. So it likes hydrophobic molecules dissolved in fats or oils. They like fats and oils, so they are called hydrophobic. That is, they have a hydrophobia, water phobia. Okay, so ligands can be hydrophilic or hydrophobic. First, let's talk about hydrophobic ligands. Hydrophobic ligands can easily cross the plasma membrane of the cell because the plasma membrane of the cell has a hydrophobic core. So these ligands or molecules can easily pass this or cross this hydrophobic core and enter the cell. So these are mostly small molecules that can cross the plasma membrane. Let's see some examples. Glucocorticoids, thyroid hormones, estrogen, testosterone, retinoic acid, etc. Now let's see hydrophilic ligands. Hydrophilic ligands are molecules that dissolve in water, or they like water, they love water, so they are called hydrophilic ligands. But the thing is, hydrophilic ligands are big, and being hydrophilic, they cannot cross the plasma membrane because it has a hydrophobic core. So it needs a carrier or a tunnel pass through the plasma membrane and get into the cell. They have difficulty in crossing the plasma membrane. So they need a carrier or a transporter or a protein that can send the signal towards the inside of the cell. Let's see some examples: vasopressin, acetylcholine, and TRH hormone. Now, signals or these molecules are sometimes released from the cell to another cell. That's the most common probability. A signal can be released from one cell to another cell, but sometimes the signals are not released. Such signals can be a DNA damage signal or cellular localization signals, which we'll deal about in detail later. So some signals are not released at all. So they are inside the cell, and such can trigger an intracellular signaling network. So some signals are released from one cell to another, some signals are not released at all, and in the third case is in which the signals remain attached to the cell surface and other cells detect it. So these are the three possibilities. Then signal molecules may require an activation, such as a proteolytic cleavage or covalent modification, etc. And how are these molecules released? They are released by membrane diffusion or exocytosis, simple as that, or cell rupture. So in the next video, we shall talk in detail about the receptors for these signals. So have a great day and keep learning.